All right, let's talk about the blueprint for life itself, DNA. This one molecule is the master instruction manual for literally every living thing on our planet, from the smallest microbe all the way up to us. So how does this one molecule hold all that incredible power? Well, let's dive in.
Okay, think about this. Deep inside nearly every single one of your trillions of cells, there's a little command center called the nucleus. And packed inside that nucleus is the complete instruction manual for building and running, well, you. Every last detail is written in the language of one extraordinary molecule.
I mean, it sounds kind of impossible, right? How can something so microscopic hold that much information? Answering that question is exactly what we're going to do. We are going to unpack the structure of the molecule that makes it all happen, DNA.
So here's our game plan. First, we'll see how DNA is packaged up as your personal blueprint. Then we're going to break it down into its most basic building blocks. We'll get into the story of how its famous shape was discovered, uncover the simple but genius rule that holds it all together, and finally we'll connect the dots and see why its structure is the key to absolutely everything it does.
Okay, first things first. Let's zoom out and look at the big picture. DNA isn't just floating around loose. It's a ridiculously long molecule that has to be organized and packed with some serious efficiency just to fit inside a tiny nucleus.
So to solve that massive packaging problem, the DNA molecule is coiled up and then coiled again and again into these super dense thread-like things called chromosomes. The best analogy is taking a super long piece of thread and just wrapping it super tightly around a bunch of spools so you can fit it in a tiny little box.
So, how is this amazing structure actually built? Well, to understand that, we need to zoom in, way in. We're going from the chromosome level all the way down to the individual bricks that make up the whole thing.
And that basic brick is called a nucleotide. You know, if DNA is the instruction book, you can think of nucleotides as the individual letters of the alphabet. The entire gigantic DNA molecule is really just a long chain of these simple units all linked up.
Now, each nucleotide itself has three parts. You've got a phosphate group and a deoxyribose sugar, and together they form the strong, sturdy backbone of the DNA strand. But the real star of the show, the piece that actually carries the genetic code, is the nitrogenous base.
And get this, there are only 4 types of these bases in DNA. We just call them A, C, G, and T. It's kind of crazy that this simple 4-letter alphabet is all it takes to write the entire story of life. But, and this is important, these 4 bases aren't all the same. They fall into 2 different families.
You've got the purines over here, adenine and guanine. You can see they have a bigger double ring structure. And then you have the pyrimidines, cytosine and thymine. They're smaller, with just a single ring. Now, this difference in size, a big one and a small one, is a critical clue for understanding DNA shape.
So how in the world did we figure all this stuff out? Well, the discovery of DNA structure is one of the great scientific detective stories of the 20th century. It had brilliant insights, fierce competition, the whole shebang. The story really kicked into high gear in the 1950s. A biochemist named Erwin Chargaff found that in any DNA sample, the amount of adenine always seemed to equal thymine, and guanine always equaled cytosine. This was a huge hint. Then, in 1953, using that rule and some crucial X-ray images from a scientist named Rosalind Franklin, James Watson and Francis Crick finally cracked the code.
What they figured out was that DNA was a double helix. The easiest way to picture it is to imagine a ladder. Got it? Now take that ladder and twist it into a spiral. That's it. It's 2 long strands that actually run in opposite directions. Scientists call them anti-parallel, and they're just wound around each other.
So, in our ladder model, the 2 sides of the ladder, the backbone, are made of those alternating sugar and phosphate groups we mentioned. And the rungs, the steps stretching across the middle, those are made by the nitrogenous bases. But what's holding those rungs together? Right? Why don't the 2 sides of the ladder just fall apart? This brings us to a super simple but really profound rule that holds the whole structure together.
The solution is just beautiful. A big base always pairs with a small one. Remember those double ring and single ring structures? By always pairing one of each type, the ladder's rungs are always the exact same width. And this simple rule is what gives the entire DNA molecule its stable, uniform shape.
So to be specific, the purine adenine, or A, always pairs up with the pyrimidine thymine, or T, and they're held together by 2 hydrogen bonds.
And the purine guanine, G, always pairs with the pyrimidine cytosine, C. This pair is a little bit stronger, held together by 3 hydrogen bonds.
And that's the rule, A with T and G with C. This specific partnership is called complementary base pairing, and it is the most important feature of DNA. It means that if you know the sequence of bases on one strand, you automatically know the sequence of its partner strand. It's a perfect match.
Okay, so why do all these little details about the structure matter so much? Because in biology, there's one golden rule. Structure is function. The shape of a molecule determines what it can do, and DNA's elegant double helix is perfectly designed for its incredible job.
And here's the absolute genius of the design. Because those two strands are complementary, the molecule has a built-in way to copy itself. The strands can be unzipped right down the middle, and each side can then act as a perfect template to build a new partner. This is how genetic information gets passed down with such amazing accuracy. But just storing the information is only half the battle. You have to be able to use it. So the DNA master blueprint gets transcribed or copied into a temporary little message molecule called RNA. Then, that RNA message gets translated by the cell's machinery to build a specific protein that goes and does the actual work.
So you have this clear flow of information. It goes from the DNA that's kept safe in the nucleus out into the cell using that RNA messenger, where it gets turned into a protein that does a job. This is literally how the code of life becomes action.
So we'll end with this fascinating paradox. When you get right down to it, DNA is a pretty simple molecule. It's just a long, monotonous chain of 4 repeating units. It doesn't do a whole lot on its own. And yet, this boring molecule, thanks to its elegant structure, holds the key to the unbelievable diversity and complexity of all life on Earth. What other secrets are still hidden in that simple code?